So let me get started.
Yeah.
So what we're going to start looking at is a bit of genetics. So doing some biology today, a bit of genetics and meiosis, because we did mitosis last week, I think. So we'll look at meiosis and some genetics today. Okay, so what we're going to do is sort of go back sort of GCSE point. What you probably can remember from GCSE is sort of somebody called Gregor Mendel, and he was a monk who would cross different types of peas with each other. So different, so he had green smooth peas and yellow wrinkly peas. So when breeding pea plants, he noticed that sometimes two plants with round yellow peas could occasionally produce offspring plants with wrinkled green peas. And so we sort of wondered how this could happen. And this is where we come to those traditional Punnett square diagrams that we talked about before in the past. And so just sort of talking through it, let's go back to basics and remind ourselves all the different parts of these. We have sort of those keywords such as genotype, phenotype. So in this situation, if he was saying he's crossing two plants that are round yellow peas, but it could produce offspring with wrinkled green peas, that is obviously here talking about the phenotype, and that is the phenotype. That is the physical appearance of the genetics. So that's how we see whatever the genes are, the alleles are that are being expressed. And genotype is actually the two alleles that we or the plants or whatever we're talking about contain. So, for example, R and Y would be a genotype. Now, we have double amounts here. Now, what you'll have often been used to seeing is one that looks like this. We talked about it on Tuesday. Let's say it's for blue eyes and brown eyes. So we've got someone with brown eyes. We've got this, and we've got someone with blue eyes. And they go for a genetic cross, B, B, B. B, B, and what you would end up with is this. So we've got dominant and two recessives. This one, the phenotype, will be blue because we've got two recessives, and you have to have two recessives present for the phenotype to show the recessive type. This phenotype here would be brown eyes because we've got the dominant phenotype. Now, this one would be, the genotype would be homozygous recessive. Homozygous because both of the alleles are the same, and recessive because that's the type of gene that the blue gene would be. Here we'd have brown, and brown would be heterozygous because we've got two different alleles, heterozygous, dominant. You can also have heterozygous, you can also have homozygous dominant, sorry, so you could have homozygous dominant, and that would look like this. Oh, but obviously we haven't got that in this cross at the minute. So the possible outcomes that you could have from this genetic cross is, again, heterozygous dominant. You could have homozygous recessive, heterozygous dominant, and homozygous recessive. So it'd be a 1-to-1 ratio chance of getting one or the other. And the phenotype for these would be brown and blue. So these are our genotypes from the parents. Genotype would be here, and then the phenotype would be that it's blue. Now, let's clear all this a minute. Here, it's a little bit more complicated. We've got an extra 2, 4, 8, 12 squares in our Punnett square. And that's because actually we're talking about a genetic cross here that involves two different genes. So we've got R and Y. So R for round, I think. So yes, so we've got round yellow seeds here. So if we have a look, this is our this one would be classed as homozygous dominant for both. So it would be homozygous dominant. So R here, this one is for round. This one is for yellow. This one is for green. This one is for wrinkled. So when we're crossing them and we've got a whole different range of genotypes, what actually happens is, let me just get rid of that because we don't need that there actually, is we end up with more than one different combination involved. So because they are co-dominant, they occur together, we're going to put them together. So if it was a heterozygous individual like this one is, RR, recessive dominant, we have 4 different possible genotypes that we could have. So we could have dominant, recessive dominant, dominant recessive, and recessive recessive. So we have the same on both sides. So we come up with this Punnett square. And we call this Mendelian genetics. And what we should find is if we cross a heterozygous individual with another heterozygous individual, we should get a Mendelian ratio of 9, 3, 3, 1. So we should get 9, and that is our phenotype. Okay, so that is our phenotype. So we should get 9 that are yellow and round. Well, we've got 1, 2, 3, 4, 5, 6, 7, 8. Oh, missed it. 9. Now they don't all contain the same genotype exactly. This one is all dominant. This one's got a recessive individual in. This one's got another different recessive individual in. This one's got 2 recessive individuals, alleles. This one's got 1 recessive allele. This one's got 2 recessive alleles. 1 recessive allele, 2 recessive alleles. So this Mendelian ratio applies to phenotypes. So that is our 9 yellow and round. Then we say there should be 3 green and round. Yep, 1, 2, 3. So green is recessive. So they will have to have both the genes for the allele for being recessive need to be present. So they're both present. But actually, for being round's dominant. So actually 2 of them are carrying a recessive allele for that as well. The next set is yellow and wrinkled. So we've got 1, 2, 3 there. And this time being wrinkled is recessive. So we need to again have that recessive there. This one's yellow is dominant. So it doesn't matter if it's carrying 2 dominant or a recessive, as long as we've got those 2 recessive alleles to make it wrinkled. This one's carrying a recessive individual as well. And then finally, 1 green and wrinkled. And that's because they all need to be recessive there. Okay, so we call these Mendelian genetics and the Mendelian ratio. That's what we're going to look into, a little bit more in a second. So, and this is what would happen if we cross something different as well. How we would get to this is we sort of looked at this first part. If we had round yellow seeds, what we could actually get is if we had pure breeding round yellow seeds and we crossed it with pure breeding wrinkled green seeds, they could be crossbred together and we get this heterozygous individual. So the parents are homozygous, dominant and homozygous recessive. We cross them, our first generation, we get a heterozygous dominant individual, and then we get this second generation, and these are our potential ratios for our second generation, which is our Mendelian ratios of 9, 3, 3, 1. Okay, so what we're going to look at is genetic inheritance and meiosis. So we're going to know the meanings of the terms gene, allele, genotype, phenotype, recessive, dominant, incomplete dominance, homozygote and heterozygote. So lots of those words you'll remember from GCSE. They're not new. Incomplete dominance is a new term that you won't have come across before. And then we'll sort of work from there. And we're also going to look at the patterns of inheritance, interpretation of genetic pedigree diagrams. So they're slightly different to the Punnett squares that we've just gone through, and in the context of monohybrid inheritance. So A star, you need to predict outcomes of genetic crosses and pedigree diagrams. B grades, understand relevance of meiosis and inherited traits. C, understand the meaning of keywords in genetic inheritance, be able to draw a monohybrid genetic cross diagram, and understand how meiosis ensures variation. Prior knowledge, you should know that genes have more than one form, and these are called alleles, like we've already discussed. So we have potentially at least 2 different forms of it, maybe 3, 4, 5, it just depends. You should also understand the meanings of the terms homozygous, heterozygous, recessive, and dominant. So we're going to have a quick look at some chromosomes. And so here we go. So the locus is, these are some really important actually. Oh, here we go. I'm going to go through these key terms with you first and then go through them. So these are some of the words that you will have heard of before, some of them you won't have. But one word that you definitely probably won't have heard of before is probably locus. All the others you should have come across. So we'll go through locus really quickly. Locus is just the location of a gene on a chromosome. It's quite a nice way to remember it. Is locus sounds like location. So the location of the gene on a chromosome. Recessive is an allele that only shows its effect on the phenotype when the dominant allele is not present. So that's important to remember. Dominant is an allele that shows its effective phenotype when 2 different versions of an allele are present in the genotype. Obviously, it can also still show if 2 of them are the same if they're both dominant as well. Gene, a length of DNA that codes for a polypeptide. A genotype is a type of alleles or genes that the organism has. So, for example, it can be homozygous, heterozygous, whereas the phenotype is a characteristic caused by the genotype or the observable effects of genotype, i.e. the colour of your hair, the colour of your eyes. Heterozygous, when an organism has 2 different copies of an allele. Alleles are the different versions of the same gene. So, for example, the gene that codes for your eye colour, you can have variations: blue, brown, black, green, hazel, everything on the spectrum of different eye colours. And then finally, homozygous is when an organism has 2 identical copies of an allele. So we're just going to, if this would move. Let's get rid of this. So look at some more keywords for genetics. So we have got 2 homologous chromosomes. So 2 homologous chromosomes. So remember, chromosomes come in pairs, and they are pairs of the same chromosome. Now, it doesn't mean that everything on them are identical, but they are. We have a pair for chromosome 1. We have 2 for chromosome 2. So you're getting the idea here. So these are our gene locus, for example. So this is where our gene is located. A is for our allele. Now, it's different on both of the homologous chromosomes. That is showing the potential that on the chromosome on the left, it is dominant, it carries a dominant allele, but on the chromosome on the right, it is carrying the recessive allele. They could both be the same, but we're just sort of representing here that those alleles can be different. Here, for example, these 2 alleles on this chromosome are homozygous. Obviously not above each other, so not the yellow F and the red F, but actually looking across at the 2 chromosomes, and it's homozygous here for these 2 alleles, homozygous here for the red 2 alleles. The bottom is heterozygous because we've got 2 different alleles there, such as it was up in this allele here. And then we have the centromere. Remembering the centromere is that central part of the chromosome when we're going through mitosis, they go and bind at the centromere and they make that like cross shape, and then that's where they get pulled apart. So we sort of had a little chat about cystic fibrosis the other day, so I'm just going to have a little bit more about cystic fibrosis. So cystic fibrosis, those symptoms where we have that thickened mucus, is brought about by the non-functioning of the CFTR protein. The CFTR protein is coded for by one gene, and there's only two different alleles for this gene. F, so our dominant codes for the normal function CFTR protein, and F, recessive, codes for the non-functioning CFTR protein. Capital F is dominant to recessive F, as indicated by the capital letters. So your three possible genotypes and phenotypes you could get are homozygous dominant, so your phenotype would be normal. Homozygous recessive, your phenotype would be cystic fibrosis. Heterozygous, your phenotype again would be normal, but you are a carrier of that recessive allele, but obviously that does not actually affect your phenotype at all. So we're going to have a recap of basic genetics. So phenotypes, if we're looking at these individuals on this picture, we've got these chickens, our phenotypes would be red or white. Our genotypes in this situation can be homozygous dominant, heterozygous, or homozygous recessive. Our dominant allele then is capital R, our recessive allele is lowercase r. Heterozygous are dominant and recessive. Homozygous can be either both dominant or both recessive. So we're going to look at a pedigree analysis. So I'm just going to talk through, I'm just going to see if I can make this pedigree analysis actually a little bit bigger. Yeah, there we go. Oh, I haven't enabled editing, that's really annoying. I'm just going to make that a bit bigger so we can see that better. Basically, while I'm waiting for that to open a bit better, a pedigree analysis is, it's a little bit, it's almost like a family tree. So we'll work on sort of family tree basis, and we're using the word pedigree there, and if you might have heard the term pedigree referred to oh no, I didn't want to do that. When people talk about dogs, they say, oh, I've got a pedigree dog. What that means is they've got a dog where they actually just know its lineage, know where it's come from. So we can do a pedigree analysis of humans as well. So it's basically like, it's almost like a family tree. So if we look at this, it's basically showing 3 generations of a family tree without people's names on. So first generation, we're showing, well, in total, we've got 4 individuals in the first generation. We've got a male, a female, a male, and a female. These are unaffected individuals, so they're unaffected of cystic fibrosis, for example. This couple go on to have 1 child who is male, who is unaffected by cystic fibrosis. This couple also go on to have 1 child who is female, who is unaffected from cystic fibrosis. They go on and have 6 children, for example. They have 6 children. 4 of them are unaffected. We have a female, a male, and a male. But they have 2 individuals who are affected. So they have female and a male who are affected with cystic fibrosis. So what comes into this situation is we start to go, okay, so this pedigree analysis shows us who has or hasn't got a disease. What it doesn't show us is where this individual and this individual inherited it from. Because obviously if it's a recessive, if it's a homozygous recessive disease, and that means that they must have got it from up here somewhere. And this pedigree chart doesn't really clearly show us that. So what we're going to do is we're going to look at it here. So if we did this, okay, so which individuals suffer from the disease? Well, we can see straight away. I'm going to draw with this pen. The individuals 2 and 3 suffer from the disease. We can see that really clearly using the key. 2 and 3 suffer from the disease. You can get given a pedigree analysis in an exam where you get asked questions like this to analyse them. So it's really important to know the ins and outs of them and understand what they're asking. Okay, you can ask what do the squares and circles mean? Well, squares and circles mean squares are male, circles are female. How can it be that 2 people in generation 3 have the disease when nobody in generations 1 and 2 had it? Well, if 2 people in generation 3 have got it, what this really suggests, and this is the important word, it suggests is that the disease must be homozygous recessive. Because obviously they've inherited the gene from somewhere for this disease. These 2 have inherited the gene somewhere for this disease up here, but none of these people are sufferers. Therefore, up here, we must have carriers. And if you're going to be a carrier, that means that it's going to be a recessive gene, which means to then be a sufferer or affected by it, you'd have to be homozygous recessive. Oh, okay, so what genotype does a carrier have? So a carrier is always going to have a genotype. It was going to be heterozygous. So all carriers are going to have, so let's say, yeah, so all carriers are going to be heterozygous. Let's use those Fs that they were doing earlier. So they would be FF for their genotype. So who could have been a carrier? Okay, well, that's quite interesting. Well, let's have a look. We know that mum and dad must have been a carrier. And so we think, okay, well, why do we know that mum and dad must have been a carrier? Well, mum and dad must have been a carrier because they need to have inherited 2. So 2 and 3 needs to have inherited 2 alleles, 2 recessive alleles, which means that because these 2 are unaffected, they would have to carry a dominant each. But for these 2 individuals to inherit them and become recessive, means they have to have inherited 1 from both mother and father, so 1 and 2. So we know this person is a carrier, this person is a carrier. We then know that either 1 or 2 is a carrier, and we know that 3 or 4 is a carrier for the child number 2 to be a carrier and for individual number 1 to be a carrier. Is this a dominant or recessive disease? Well, it's clearly a recessive disease because neither of the first or second generation are affected, yet we have individuals in the 3rd generation who are affected, so that insinuates that this is a recessive disease. How do we know? So we've just explained that through the workings. So if it is a recessive disease, can one parent who is a carrier and another with a normal alleles produce a child with the disease? Okay, so you can even just draw one out quickly to work that out. So let's say we've got a parent who is a carrier and a parent with normal alleles. Can they have a child who has the disease? FF? No, 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 no. So these would all be unaffected, but these 2 would be carriers, but they are not affected. So you cannot have a parent, you can't have one parent being a carrier and the other one being normal producing a child with disease. So we know up here in generation 1, one parent has to be a carrier and one parent could be normal. One parent has to be a carrier or the other could be normal. Now I'm using the word could because actually both could be carriers, but it would just happen that they only inherited the one recessive gene from one of the parents. Okay, so how many people in generation 1 had to have the carrier genotype? Which sides? So in generation 1, we have to have at least, 2 people in generation 1. 2 people in generation 1 have to have a carrier genotype. It could be all 4, but it has to be 2. Which sides? Well, we have to have at least 1 on this side, and we have to have at least 1 on this side. So we need a carrier on both sides. Ooh. So dominant diseases. So I'm going to do a quick genetic cross for these parents. I'm going to show the percentage probability of the offspring disease. The disease allele is dominant. So you can get shown these a lot and asked to do these. This would get you 4 marks in exam. So an exam tip would be to remember to show the parent genotypes, offspring genotypes, which offspring have the disease, and the probability. So this is a dominant disease, so this is quite interesting. So this is where you suffer from disease, and you would be need to either be heterozygous or homozygous dominant to have the disease. So I'm hoping I've wrote it on here. Oh, right. So I'm going to write it out separately. So genetic cross. Here we go. So we've got one parent, HH. I'm just going to write parents here. Other parent, HH. That's their genotypes. So show the parent genotype. I'm going to show the offspring genotype. So we could get H with H. We could get H with H. I mean, me saying H isn't really that helpful. We could get recessive with recessive. And we could get recessive with recessive. So parent genotype, these are our offspring genotypes. Which offspring have the disease? Okay, so which offspring would have the disease? This offspring would have the disease. This offspring would have the disease because it is a dominant disease. And the probability, will it be a 1 in 1 chance? 1 in 1 ratio, sorry. So so you've got a 50% chance. So you've got a 50% chance, 1 in 2 chance of having the disease. Marfan syndrome is also a dominant disease, so it'd be actually quite interesting to look into as well. So let me clear that, and I'll show you the proper drawing for it. If this work. Here we go. So yeah, parents, like I've just shown you, offspring with disease, Huntington's allele and normal allele. So we use Huntington's here. So here's another pedigree chart. Have we got any? Okay. So here we've got another pedigree chart. So what we've got here is we've got male and female. Male and female are always squares and circles, and the coloured in one is always affected individuals. So we've got affected individual in the 1st generation with someone in the 2nd generation. They have 1, 2, 3, 4, 5 children. 3 of them are also sufferers. 2 of them aren't. So start to have a think what this might suggest. So let's go through some examples. Let's say this was a dominant disease. Let's go through a dominant disease idea. Okay, well, dominant disease idea. Yeah, no, that could work. So that one could be HH. That one could be HH. It's about 50-50 chance they've had. How many kids did we have? We had 5 kids, so around that ratio. So it could be that. Could it be this ratio? Could it be this combination as well? Let's have a look. Could it be homozygous recessive? So we've got HH. And could this person be homozygous dominant. No, we know they can't do that because then none of them would be sufferers. So we know it can't be that. Could this individual be heterozygous? Yeah. But we wouldn't have as high a ratio of individuals suffering because we would have so yeah, and again, we'd have 50-50. So it could be either way for them. So these ones would be like that. So depending on which way we're going to do this. So look at the third generation. Okay, so none of the individuals that people have had children with are sufferers. This is quite an interesting one to look at because neither the child from parent 1 and 2 up here and their a partner who's female have 3 children and none of them are sufferers. So if none of them are sufferers, that means that we've definitely got an interesting situation here. So again, we've got here sufferers, but with non-sufferer, non-sufferers with non-sufferers, and sufferers. So what this is possibly indicating is that maybe it's a dominant recessive homozygous dominant disease, and it could still be homozygous recessive because again, if we look at our ratios, homozygous recessive, if they were a carrier and they were a carrier. So everyone that they have to have had children with would all have to be carriers. It's a potential. Chances are it's not very likely. So actually, the chances are it's more likely to be a dominant disease. What we're going to look at now is a sex-linked disease. If I could get this page to move on. Here we go, sex-linked diseases. Okay, so yellow identifies carriers. So yellow identifies carriers. Blue identifies affected individuals. So what we have up here is a sex-linked disease carried by a female and the individual male and a male who is normal, is not affected by it. They then have a daughter who is a carrier and a son who is a sufferer. Now, why? Again, if we look straight away, we've got more males who are sufferers. We've only got one female who's a sufferer. Why? Well, it's to do with our chromosomes. So females carry XX chromosomes, male carry XY. If this is a gene that is carried on the part of the X chromosome that is not found on the Y chromosome, it's found on the X chromosome, means it's then not found on the Y chromosome. So the males will only inherit the one. So for example, this female, for example, is a carrier, so she has this XP here. So whilst this boy's inherited the XN, which is a normal, and the Y from his father, this individual's inherited the XP, which is the one with the disease. Now, because their other allele is Y, they have no backup. And so that it doesn't carry a backup allele for this disease. So if it's on the female X chromosome, if the disease is found on the X chromosome and you carry it, then you will have it. Whereas for a female, because they have a backup allele, then they will just be a carrier. But it does mean that the female can eventually inherit it, such as here, because she then inherits the XP from that affected parent and from that carrier parent. So if I just clear all my scribbles off straight away, so we've got affected individual female. She passes it on to a carrier daughter and a son who is affected. A son who is affected has children with a female who is unaffected. They have 2 carrier females. Males cannot be carriers because males do not have that second X chromosome, so they can either only be sufferers or normal. This way, that individual has children with a sufferer, therefore they have 1 female who's a carrier, a male who's normal, a female who's a carrier, and a female who's a sufferer and a male who is a sufferer as well. So they work quite differently because they do not have them back up. So yes, so let's just clear all my scribbles again. Sex of that individual is female. This person's phenotype, well, we would say the phenotype, they are affected, they have the disease. What do we think the N and P stand for? Well, the N and P stand for potentially normal or problematic. They stand for the different alleles. Is this recessive or dominant disease? Okay, so this is an interesting one to look at, but clearly it is a recessive disease, because if we look here, the females only have a disease when they carry both P's. They carry P and N, then they are just a carrier, so that's homozygous. They're heterozygous there, so only homozygous recessive females have the disease. How would you describe this person's phenotype? This person's phenotype would be heterozygous, and the phenotype would be that they are a carrier. How would you describe that person's genotype? They're heterozygous. Okay. Mitosis and meiosis. So, mitosis was the splitting of a cell. We're now going to look at meiosis. So meiosis begins with a single cell. This is our original cell. We have 1. The cell makes a copy of each of its chromosomes. And now there are 2 stages of cell division. We have our first stage of cell division, and then it goes through cell division again a second time. Now, between the first and the second division, there is no replication of any chromosomes. That's our major difference in mitosis. Obviously, here we're talking about gametes. We're talking about sex cells. So those 4 new cells, those 4 daughter cells at the bottom, they're going to have half the amount of the original chromosome, half the amount of chromosomes as the original cell had. So how many chromosomes do the new cells have compared to the original? They have half the amount. So meiosis produces 4 new sex cells, and they have half the original number of chromosomes. In animals, meiosis produces ova in females and sperm cells in males. In plants, the gametes produced by meiosis are pollen grains and ovules. Mitosis is also called reduction division. So reduction division is, we're not just dividing, we're reducing the amount of chromosomes. So the reduction part of a division is actually the fact that we're reducing the number of chromosomes. So we're actually reducing the numbers there. So how does meiosis turn 1 cell into 4 new sex cells? We go like this. So so we go from 1 cell into 4 new sex cells. So the first stage is, oh no, press it the wrong way. We go through cell replication, DNA replication, and so all the chromosomes are replicated, including our spindle fibers, our centrioles at either end of the cell. So the first part is very similar to mitosis because we duplicate the chromosomes. The chromosomes align in the middle. The spindle fibers attach at the centromeres. They get pulled to either side of the cell. The cell then goes through cytokinesis, and we have 2 cells. 2 cells that have the same amount of DNA as the first cell. Okay, same amount of DNA as the first cell. Then they go through cell division again. They line up along the middle of the cell. The spindle fibers attached to the centromeres. The centromeres pull the chromosomes apart to either side of the cell. The cell then splits. Cell then splits, goes through cytokinesis, and then we get 4 separate cells. This is a really important stage here. The fact that we don't have a replication here. That means that none of these cells are identical. And that's really, really important to meiosis. Meiosis is the production of 4 non-identical cells. 4 non-identical cells, that means that we're increasing, what's the word I'm looking for? We are increasing variation. So our variation has increased, and that's really, really important in meiosis. Mitosis, we want clones. We want everything to be exactly the same. We don't want a skin cell to have any variation in it. We want it to work and act exactly the same as the skin cell that came before it. Meiosis, we don't want all of the gametes to look identically the same. If they did, and gametes were produced using mitosis, then every single individual, every offspring from both parents would be identical to each other. So whilst you would still have half your genetics from the mother, half the genetics from the father, that offspring's siblings would all be genetically identical to itself. Therefore, meiosis allows them to have each offspring to have a different genetic makeup from each parent. So each of the half from the parent is a different half every time. So I'm not going to do this. So, So this is a really quick recap of the differences between mitosis and meiosis. The number of cells needed at the beginning is 1 for both of them. Mitosis makes 2 cells, also referred to as daughter cells, whereas meiosis makes 4 daughter cells. So the cells at the start of mitosis and meiosis are called diploid cells. They have that full set of genetic information. By the end of mitosis, the end cells, the 2 daughter cells, are each diploid. They each have the same amount of chromosomes as that one original cell had. Whereas in meiosis, they become haploid cells. Think of and half. They have half the amount of chromosomes as that original cell had. So mitosis is used for repair, growth, asexual reproduction, whereas meiosis is only using gametes. Mitosis, does it involve crossing over chiasmata? No. Meiosis, yes. And that's what we're going to go have a look at now. Are the cells produced genetically identical or different? Mitosis are identical. Meiosis, they are different. Crossing over the chiasmata. Can I get an image of this anywhere? Here we go. So this is quite like a nice overview of mitosis and meiosis again. So, parent cell, before chromosome replication, 2n equals 4, okay? So, in mitosis, we have prophase, we have our duplicated chromosomes, we go through metaphase, the chromosomes align at the metaphase plate, anaphase and telophase, sister chromatids separate during anaphase. Therefore, we get cytokinesis, we get 2 new daughter cells that both have 2n, and we have them there. Meiosis, meiosis is a bit different. So meiosis, we have meiosis 1 and meiosis 2. So meiosis 1, in prophase, the chiasma, the site of crossing over, is the site of crossing over, and we have a tetrad formed by synapse of homologous chromosomes, and we have crossing over start to occur. So crossing over is basically where a bit of one chromosome swaps to being on a bit of another chromosome, is basically adding genetic variation in there. So not only are we just pulling apart different chromosomes, having different mixtures of them in each one, that would have somewhat genetic variation, but not a huge amount. So we actually have crossing over the chiasmas and meiosis to increase this genetic variation. So that's in prophase 1. In metaphase 1, the tetrads align at the metaphase plate. Now, we call them tetrads at this point, just because we've had some crossing over. Then we have anaphase and telophase 1. So our homologous chromosomes separate during anaphase 1 and sister chromatids remain together. So actually our chromatids are staying together. So they're splitting apart and they're staying together. Then meiosis 2, we have no further chromosomal replication. Sister chromatids separate during anaphase 2. We have no further crossing over. So it's just more splitting of them apart. And here is a diagram in meiosis of what's happening here. So, so we have random assortment and the chiasmata. So here we go. We've got mitosis, and we have this crossing over. So that adds to that random assortment. So here we have two that are randomly assorted. So then we get our two separate cells. They go through meiosis again, and then we get 4 separate cells with half the genetic information as the original cell, and we've actually had some crossing over to get some random assortment inside there. So, oh, nice. Okay, so I'm going to leave that there. That was a bit of a whistle-stop tour of genetics. What I'm going to do is, I know I haven't, oh, actually, let me just pause the recording. We don't need me rambling.